Hi guys, Anna is here, and in this A-Level Biology Masterclass I cover topics less than in 10 minutes, so please grab your paper and we can get into this topic. This video will focus on recombinant gene technology, and it will be the first video, so we're going to be talking about how to isolate DNA fragments. And it will be separated into three technologies. But first of all, let's have an introduction of recombinant gene technology. Recombinant gene technology is the process of making a transgenic organism. Transgenic organism means that there'll be different DNAs from different organisms, usually two, that are combined into it. And eventually, that means that that transgenic organism will have a recombinant DNA. A classic example is, and the one we're going to use in this video, is human insulin gene, which is combined and is going to be inserted into bacterial plasmid, and which will be the focus of this video. So, for example, the human insulin gene will be isolated from a beta pancreas cell from the human, and then the bacterial it will be inserted into the bacterial plasmid, which is represented in green here as a separate DNA. And eventually, once the insulin gene and the bacterial plasmid are combined together, that plasmid we can call a recombinant plasmid. The reason for using recombinant gene technology is to make, for example, these recombinant plasmid, which we then put back into bacteria and which can be used to produce a certain protein such as insulin here on a large scale. So in this video, we're going to focus firstly on how to obtain a desired DNA fragment. Again, you could keep human insulin as an example in your mind in this case, but basically there are three different technologies. First way to obtain a DNA fragment is by cutting it out using restriction endonucleases. Method number two involves using an enzyme called reverse transcriptase, which will produce DNA fragment from messenger RNA. And then the technology number three includes using the apparatus called gene machine, which is more rarely used, but it's producing the DNA fragment completely de novo, so completely from scratch. And so the focus of this video will be comparing and contrasting those three technologies. So let's get into it. So the first technology includes using the enzymes called restriction endonucleases to cut a certain DNA fragment of interest. So it's important for A-levels to call these enzymes restriction endonucleases rather than restriction enzymes, because some boards actually don't accept restriction enzymes anymore, because restriction endonuclease is a proper name for it. I'm going to draw a section of DNA, but in this case, obviously, I can't draw 2 meters of DNA here. So I'm just drawing a DNA with particular insulin gene presented here. And what we need to do now is to use two different restriction endonuclease enzymes. And these restriction endonuclease enzymes will be selected to cut certain sequences on both sides of the insulin gene. Those sequences are called palindromic sequences, and they have to be different. So palindromic sequences, they are the mirror images of the sequences of themselves. So to explain it in a slightly better manner, I'm actually going to give you some examples. So it's going to go GAA-TTC, and then the opposite strand of the DNA, complementary one, is going to be CTT-AAG. And as you can see in this example, those both sequences are the mirror images as you read them in both directions. So a similar case we're going to do is on the other side. So a different palindromic sequence could be TGC and GCA, and as you can see on the opposite side, it will be read in the same direction. So the one restriction endonuclease will cut one palindromic sequence, and then a different restriction endonuclease will cut a second palindromic sequence. And using both of those enzymes will cut out an insulin gene out of the entirety of the DNA within a particular cell. And what we're going to do now on the next page is to show how this process happens. So here we go. I've just copied it over. So as a reminder, we're going to have restriction endonucleases, which I'm drawing here in different colors, that will cut the palindromic sequences on each side of a particular gene, being an insulin gene in this case. And what will be left over of that is obviously the insulin gene is going to be in the middle, but the way the restriction endonucleases are cutting that particular fragment is by leaving sticky ends on each side of the insulin gene. So those sticky ends, they're single-stranded overhangs of DNA, which will be used then as fragments, almost like jigsaw puzzles, to combine them with the plasmid of a bacteria. And this is why, and it's one of the most important exam points, guys, is that you need to state that the same restriction enzymes will be used to cut the plasmid. It's important to note that some restriction endonuclease enzymes are able to also cut and produce blunt ends. So when the DNA is cut, it basically produces like a straight line, as I'm representing here in pink. And basically, those blunt ends are not useful for recombinant technology, and therefore we're interested in restriction endonucleases producing sticky ends. This brings us to technology number 2, where we're going to be discussing how reverse transcriptase as an enzyme is used to produce our fragment of interest. So to give you an example, in some cases, scientists might not have DNA available for one reason or the other, but they might have cells that produce loads of messenger RNA for that particular gene. So what we can do in this case, or what scientists could do, is basically extract messenger RNA from a particular cell. So if we're using insulin gene as an example, we might be able to extract mRNA from pancreatic cells. So in this case, we start with a single-stranded mRNA, and then the enzyme called reverse transcriptase will be added, which will produce a complementary copy of messenger RNA called cDNA, which stands for complementary DNA. So this is great. We have a copy of DNA. However, that fragment actually has now one strand, which is messenger RNA, and another strand of cDNA. So therefore, we have to continue and actually get rid of the strand of mRNA, which we used originally, by an enzyme called RNase, which will just digest and remove that messenger RNA strand, and we will be left with a single-stranded cDNA in this case. Again, that's not enough because we need our DNA to be double-stranded. So in turn, we're going to add an enzyme called DNA polymerase to synthesize a second complementary strand of DNA, as I'm just drawing here. And voila, this will give us the final strand of DNA or DNA fragment, which will be like a DNA fragment for human insulin gene, for example. And guys, if you can learn this kind of doodle to understand the reverse transcriptase, I'm hoping that this will simplify this massively for you. And this is everything you need to know for reverse transcriptase. So the final technology is called gene machine. And I'm going to be honest, not all exam boards require you to know this. AQA definitely does, but I'm not sure about the rest. So you will just have to check your own syllabus, but also this is good for your extension knowledge. So gene machine is called a de novo method. So it means that we're going to be building DNA from scratch that didn't exist before. And the way it works, a known DNA sequence will be inputted into the apparatus, into like a computer, and then that apparatus will build a completely new DNA molecule or DNA fragment of our interest. So here I'm trying to do a 3D shape of the matrix, which will be one part of the apparatus. And so imagine this kind of flat matrix or like a flat platform that will be used to grow short sequences of DNA upwards, like little strings. So those little molecules that I'm drawing, so each circle represents a single nucleotide, which, reminder guys, is a monomer of DNA. And then the whole sequence, that is might be 5 to 12 DNA nucleotides, is called oligonucleotide. Oligonucleotide, or like that part of oligo, means that it's a relatively short sequence, because you guys learn about monomers, which is single kind of molecules. Then you might learn about two molecules joining together and then going all the way to the polymers. But in this case, oligonucleotides means something relatively short. In this case, the nucleotides are single-stranded. So because we can't grow them too long on the matrix, so the single-stranded DNA molecules are grown on the matrix, then they will be taken off, they will be joined to make them into longer strand, and then they will be joined to make them into a double-stranded DNA. Thank you so much for watching this video. I hope this simplifies the recombinant gene technology a little bit more, and I will be releasing more videos in this series as soon as possible. Please press the likes and subscribe for my channel, and I'll see you in my next video. Thank you. Goodbye.